We will discuss about immunoprophylaxis. Now, till now, I think we have discussed about the human immune system, how the immune system works, and how it helps us preventing various infections, to acquire the infections, and how to prevent the disease from happening. Now, in this context, we'll try to understand what immunoprophylaxis is, and we'll also go through the journey of this picture. If you are seeing over here, this is the picture that shows the first development of vaccination as depicted for, by কাদেরকে রাখো বলছি কাদেরকে রিপ্লেসমেন্ট দিয়ে যাও আসার নাম নেই খোলার নাম নেই সবই যদি করতে হয় তাহলে আর কি বলবো বলো at today's status of vaccinations. First of all, look at this graph. This is the graph which shows the median life expectancy of human beings from 1500 to 2016. And I think you can appreciate that the mean life expectancy has increased exponentially in the last 100 to 200 years. Now, one of the biggest reasons of increased life expectancy is, if we look at the causes, a big chunk is because of the prevention of enteric and infectious diseases. So, disease prevention has actually contributed a lot for increasing the life expectancy over the last 200 years. Now, if you look at the history of the development from 1400 to 1700s, obviously the people were not aware of the various causes of the diseases, but it was well established that people who had already got the disease were not getting the disease the second time, and hence that led to the foundations of the immune reaction. Coming back to the year 1796, Dr. Edward Jenner first demonstrated that if he inoculated a particular person with the fluid extracted from the lesions of cowpox, that person did not get smallpox again. So that was the very first foundations of vaccination that was done with evidence. Thereafter, the various powerful people started implementing the vaccination. Now, keep in mind, at that point of time, smallpox was a very dangerous disease capable of almost cleansing the human population. Moving ahead to the year 1985, Louis Pasteur first successfully developed a vaccine that could be given to prevent rabies infection. Till that time, rabies was pretty much a disease which cannot be prevented, hence that led to the breakthrough. Following the principles of making the anti-rabies vaccines, more vaccines were developed against various pathogens, be it anthrax, cholera, etc. A lot of vaccines were developed. Some were successful, some were not. Next, in the early 19th century, the first thing that happened in the 1918 to 1919 is called as the Spanish flu, a variant of influenza that spread across the Europe and the Americas, and a huge number of people died. That led to further more dedicated research work in vaccination. Over the years, many more vaccines were developed, but worldwide effort was started in the year 1967, where the WHO, or the World Health Organization, initiated the massive vaccination for smallpox. And after the few years, the world were able to eradicate the disease itself. And today, there are no cases of smallpox, and neither are the smallpox viruses. So this was an evidence that a successful vaccination campaign can actually prevent the disease, and in some cases it can also eradicate the disease. Following the same model, the WHO also then started advocating for vaccination against another disease which used to cause severe mortality in children, that is the measles, later on combined with both rubella and mumps. Coming back to the year 1988, WHO started mass vaccination for polio. Polio was at that point of time very much endemic, especially in the Asian countries. And mass polio or pulse polio vaccination was started even from the government of India. and at present, India has been declared as polio free. So, there lies another very successful campaign that has been done by the mankind in prevention of a disease. Next, in the year 2016, another campaign was started for mass vaccination against meningitis. And by 2019, the WHO advocated for a vaccine against Ebola virus. Now, Ebola is a virus which causes a hemorrhagic disease. After the infection, people usually bleed out to death. And it was started spreading outbreaks were happening in Africa in many countries. And WHO had successfully prevented by introduction of this vaccine. And finally, when you look at the recently past pandemic, that is the 2021 COVID pandemic, it was evidence that mass vaccination actually prevented not only mortality but also morbidity. Many types of vaccines were used in various countries. Nevertheless, those vaccines were capable of reducing morbidity and mortality. Now, having said that, let us understand these topics in slightly more detail. Now, when we say the word immunoprophylaxis, what we actually mean? That means immuno and prophylaxis. That is, try to prevent the disease with the help of the immune system. So, it is the prevention of the disease through intentional production of active or passive immunity. It encompasses both active immunity and passive immunity. Next, what do we mean by active immunity and passive immunity? Any ideas? No. No. So, the primary goal is to stimulate the immune system, and thereby we can classify it into two types, either active immunoprophylaxis or passive immunoprophylaxis. Now, if you look at human immunity in brief, the human immunity has two types of immunity. One is the adaptive and the innate immunity. And both the innate and adaptive immunity, coming to the adaptive immunity, we have the natural and the artificial. Artificial means that is given by us, the healthcare workers, or iatrogen. Natural immunity can also be active or passive. Suppose you do get an infection. Suppose you have been infected by a mumps virus and you have survived. What will happen? Your body will start producing antibodies against mumps virus, and those antibodies will remain throughout your lifetime. So you will not get infected with the same virus again in your lifetime. So that is what type of immunity? That is an active immunity. Your body has to work to develop those antibodies. Now, suppose there is a mother who had been infected by mumps virus in her childhood. She has antibodies against mumps virus, and now, uh, suppose she has conceived, and during pregnancy, she will transfer the IgG antibodies to the child in utero. So the child is protected by the maternal antibodies. The child is not producing the antibodies, but the maternal antibodies are crossing to the child. So this is the type of passive protection, but it occurs naturally. The same thing can be done artificially or iatrogenically, where we can either introduce the pathogen into the person's body so as to stimulate production of antibodies, or we can directly give the antibodies. If we are giving the antibodies directly, we call it as passive immunization, and if we are giving the vaccines, we call it as active immunization. Okay, fine. Now, how does this occur? It all started with the smallpox and the cowpox. Now, if we look at the viruses, both the viruses have almost similar structure and similar antigen. Hence, when the cowpox virus was introduced, it started producing antibodies against these antigens. And the same antibodies, because the antigens were shared, the same antibodies were able to prevent smallpox. Keep this in mind. Next, whenever we are introducing the vaccine to the human body, it will trigger the immune system. The antigen presenting cells will process the antigens, and it will stimulate both the B cells and T cells, triggering both the cell mediated immunity and the antibody production. Now, even if you do get the infection naturally, your body will produce antibodies. Then an obvious question arises, then why do we need to vaccinate? Okay. So look at this picture over here. What happens naturally? Suppose a person gets infected. After a time, the body will start producing antibodies. Now, if the patient recovers, there will be a steady state of antibody at the baseline. And if the patient gets infected again, there will be an exponential production of the same antibody the second time. This is how our body protects. Now, what will happen if we have given the same person a vaccine prior to getting infected? Now, this is the picture I think you have understood. But if the person had been given the vaccine, what will happen? The time duration for the production of antibodies will be reduced. Secondly, if based on the pathogen, that is, the pathogen has a single type of antigen, that means if the antibody is produced by the body once in your lifetime, those antibodies will work throughout your lifetime. And if there are antigenic variations, even then, the secondary rise of the antibodies, the time taken will be very much reduced. Understood? So that is why vaccination is important. Now, this is the graph. On the x-axis, you can see antigenic variability. Antigenic variability means the different types of antigen a pathogen has. And on the y-axis, we can see the vaccine development success. Let me explain to you. Suppose there is an organism like diphtheria. It has a very restricted antigens which can be expressed for production of antibodies. Hence, if a person gets vaccinated with diphtheria vaccine, the antibodies will be very much specific and will be very, very protective. On the other hand, we can see the HIV virus. The HIV virus has various different types of antigens, and many antigens can be expressed. So, a huge number of antibodies are produced. Some may be effective, some may not be effective. That is the reason why, over the period of this huge number of years that have gone by, we have seen there had been no concrete success in development of a vaccine against HIV because of the antigenic variability. However, if you see vaccines like diphtheria, tetanus, these vaccines have been developed many years ago and still very effective even still today. The main reason is because of the antigenic variability. Furthermore, these various types of vaccines, the normal thinking is that it will start producing antibodies. But it is not always the case. Why? Because it can not only produce antibodies, but some of the vaccines will also trigger the cell mediated immunity also. Like the oral polio vaccine. I think you have heard about oral polio vaccine, the vaccine that was given in the pulse polio immunization program. So it's a live virus and it mimics the natural infection. So it not only produces antibody in the humoral system but also triggers the cell mediated immunity. Based on this, now if you look at this picture, this is the total number of the vaccines that are being currently studied or under development. However, the only this little number of vaccines are actually are in use. Okay, so just to give you an idea that we use very small number of vaccines actually that have been tried, tested. Rest are all under the development. Next, very important part of a vaccine is something called as an adjuvant. So when you start reading about the vaccines, many a times you will come across a term called adjuvant. There are many types of adjuvant. A very common example of an adjuvant is alum. Now these are given along with the vaccines. What is the purpose? The purpose is very simple. These adjuvants will help in presentation of the antigen and will stimulate the immune system either furthermore, whatever may be the outcome, maybe the production of antibodies or triggering the cell mediated immunity. So basically, they accentuate the immune response. Okay. Next, what you need to understand is called as herd immunity. Herd means group of animals. So when we talk about from the community point of view, we must understand the population as a whole. So this is called as herd immunity. So if a particular population is protected against a disease, obviously Even if a single person gets the disease, that disease would not spread into the population. However, if the population is naive, a single person who gets infected can spread the disease throughout the population, as exemplified by this picture. Now, suppose there is a population in which all people are naive, immunologically naive, that means they are not immunized, as shown with the blue color. And a single person, suppose the person in the red color, gets the disease and exposes all population. What will happen? Almost all person will get the disease. Now, suppose you have vaccinated some of the person in the population. That means majority are still naive, but some do have the protective antibodies. And now this population gets exposed. What will happen? Those who have been immunized will get protected, but still the majority of the population will be affected. However, in another set of population, we have vaccinated the majority of the population. Okay, some outliers are there who are not vaccinated or not immunized, but majority are immunized. Now, if this population is exposed, most of the population will be protected. And when this population is protected, the biggest advantage is that the disease will not spread. So, I think now you have understood. So, what herd immunity means, what is the number of population that you must immunize so as to prevent the spread of the disease in that population and from that population to another population. So, there are specific mathematical models, the RO number, which have been studied in detail to give us an estimate based on the number of the people in a certain area, how many people must be vaccinated. If you go back and look what are the happenings that happened during the COVID pandemic, the government advocated for vaccination of a certain number of people. So that was the objective, to prevent the disease. Okay. Now let us move on to the next part, that is the active immunoprophylaxis. So by definition, what is active immunoprophylaxis? That is when we are iatrogenically giving the person a vaccine so as to stimulate his or her immune system to produce antibodies. Now, these active immunization vaccines can again be divided based on their valency, that is, monovalent or polyvalent. If we are targeting only a specific antigen, we call it as monovalent. If we are targeting many other antigens, we call it as polyvalent. It may be related to a particular disease or may not be. For example, pneumococcal vaccine can be monovalent against one strain only, or it can be a polyvalent in which antigens of multiple strains of the same organism, that is pneumococcus, can be given. Or we can also classify it into either homologous vaccine or heterologous vaccine. What is homologous? That means suppose we are trying to prevent pneumococcal pneumonia. So we are giving the vaccine against pneumococcus, the same organism. But sometimes there is antigenic sharing. For example, in case of tuberculosis, we are giving a different vaccine with a different strain of mycobacteria so as to prevent infection by Mycobacterium tuberculosis. That is called as a heterologous vaccines. Other than that, the main classification or main types of the vaccines would be, the first is called as the live attenuated vaccine, then inactivated or killed vaccine, cell extracted vaccines, toxoids, subunit vaccines, and finally DNA and edible vaccines. Now, what is a live attenuated vaccines? Live means the organism is still alive, it is not killed. It retains its ability to cause the disease. But again, we have another word, it is called attenuated. What do you mean by attenuation? Any idea? No. Attenuation means the virulence of the organism is reduced. So, when we inject this particular strain into a human body, it will cause the disease, but the disease manifestation will be reduced. And because it is causing the disease, it will produce the immune reaction. Understood? For example, measles vaccine is a live vaccine. If the wild strain infects you, you will have full blown manifestation of measles. But when we are giving the vaccine virus, the disease will happen similarly, but the manifestations will be very limited. So these are called as the live vaccines. The example would include, for example, in bacterial vaccines would be BCG vaccine, typhoid vaccines. Among the viral vaccines, very commonly the mumps, measles, rubella, oral polio vaccines, chickenpox vaccines, or varicella vaccines, JE, hepatitis, etc. Now, keep in mind the live vaccines, they are live. That means the pathogen can still cause the disease, but the manifestations are limited. So, this will happen only in immune competent persons only. If you give the same vaccine to an immune compromised person, what will happen? It can still cause severe disease. Similarly, in pregnant women, these live vaccines are contraindicated. However, there is a big advantage of live vaccine. Because it causes the disease by similar pathogenesis, the ability to stimulate the immune system is much better. Hence, as you already know, many of the live vaccines are given only once. So, multiple doses are usually not required. Now, suppose in the same organism, we treat the same organism by various chemicals so as to kill the pathogen. Now, it cannot cause the disease. What it only retains are the antigens. Those are called as the kill vaccines. Now, what will be the advantage of the kill vaccines? Because they are already killed, they cannot cause the disease. You can use these vaccines even in immune compromised patients as well, and even in pregnant women. The problem is because the organism is no longer viable and the whole immune stimulation is dependent solely on the antigen, we need to add various other adjuvants and give multiple doses. This is the reason why you will find most of the kill vaccines are given repeatedly and sometimes even booster doses are required. Example of kill vaccine would be like the pertussis vaccines, cholera vaccines, and in viral vaccines it will be the kill influenza vaccine, rabies vaccine, etc. Now this is important from your exam purposes. You will be asked about the differences between kill vaccines and live vaccines. So what will be the difference? Because the live vaccines are more efficient, usually a single dose suffices. But in case of kill vaccines, we need to give multiple doses. Next, adjuvants are required and duration of immunity will be shorter. Effectiveness will be shorter as compared to live vaccines because the live vaccines mimics the natural infection. Furthermore, if it is a live vaccine, we are stimulating not only IgG but also local immunoglobulins like IgA. But if we are giving kill vaccines, we are only stimulating the IgGs. A very good example would be the polio vaccine. If you are giving the kill polio vaccine, that is the injectable vaccine, we get IgG. That means that particular patient is protected. However, if we give oral polio vaccines, not only the IgG develops, but also IgA, that is mucosal immunity also develops. Now, live vaccines, obviously, it will also stimulate the cell-mediated immunity along with the humoral immunity, but kill vaccines do not. And finally, very important is that in immune deficiency and pregnancy, kill vaccines can be given, but live vaccines are contraindicated. Okay. Now, moving on, there are some diseases in which after the organism causes infection, the organism produces various toxins. The disease pathogenesis is mainly based on these toxins. So, to prevent those toxins from acting, we need antibodies we can neutralize these proteins, these toxins. So, the vaccines which produce antibodies which can neutralize the toxins are called as toxoid vaccines. Namely, diphtheria, pertussis, tetanus, all these produces respective toxins. If we give these toxoid vaccines, the body will start producing antibodies against respectively diphtheria toxin, pertussis, and tetanus toxins. So when the person gets infected with the disease progresses, but the manifestations of the disease is subdued because even if the toxin is produced, the body can neutralize the toxin. Okay. Next will be the extracted or cell fraction vaccines. In some of the organisms, they have high genetic variability. So, we need only a small part of the cell of that pathogen which can actually stimulate the immune system. Those are called as the extracted or cell fraction vaccines. Like the pneumococcal vaccine. It could be the pneumococcal capsular vaccine or the pneumococcal polysaccharide vaccine, whatever it may be. The next type of vaccine is we call as subunit vaccines. Now, what is a subunit vaccine? Subunit means we are only introducing a small part of the pathogen. Now, let us take an example. The hepatitis B virus has a protein which surrounds the virus particle. That is called as the surface antigen. Okay. Now, this helps the virus to enter into the cell and thereby causing the disease. If the body produces antibody against this antigen, the antibody will attach with the antigen and the virus cannot enter into the cell, thereby protecting the human being from getting infected. Okay, fine. Problem is that if we do introduce the virus as a whole, the person will get the disease. An easier alternative would be only to take out the surface antigen and inject it. Now it becomes very difficult. So, with the help of the biomedical engineering, what is done is that we take out the gene which codes for the surface antigen, and that gene is introduced into another organism, for example, a bacteria or an yeast. Now, this bacteria and yeast will start producing the same protein which mimics the surface antigen, and this protein is introduced. Hence, it is called as the subunit vaccine. It is extracted by the recombinant DNA technology. Similar example would be the human papilloma virus. Combined vaccines can be given. That means multiple vaccines can be combined with each other. A very common example would be diphtheria, pertussis, and tetanus that is given simultaneously, or we can give a pentavalent vaccine in which includes the DPT vaccine along with Haemophilus influenzae B and hepatitis B virus. So this is called as combined vaccination. Coming to the recent development, DNA vaccines, or as I think you have already heard about, mRNA vaccines that were given during COVID pandemic. These vaccines, how does they work? A particular nucleic acid is introduced into the body. This nucleic acid gets incorporated into the human cells and start producing a particular antigen. And this antigen will produce both antibodies for humoral immunity and cell-mediated immunity also. And the last development is called as the edible vaccines. Now, edible means something we can eat. Again, with the recombinant genetic technology, the antigens or the pathogenic material is introduced into the edible items like potato, bananas, etc. So that when a person eats, the person is exposed to these antigens and the immune system can be stimulated. The next topic is passive immunoprophylaxis. Till now, we had discussed about active immunoprophylaxis. That means vaccines are given so as the person produces the antibody. Then, what is the requirement of passive immunoprophylaxis? Passive means we are giving the antibodies directly to the patient, not the vaccine. Now, what is the requirement? The first requirement is, suppose a person is bitten by a rabid dog. We can vaccinate the person previous to the bite, then it can protect him. But suppose a person who has not been vaccinated gets the bite, what we have to do? We have to protect that person immediately. For that, we need to give the person immunoglobulins. So when we give the person immunoglobulin, we call it as passive immunoprophylaxis. Similarly, suppose a person gets the disease before the disease is manifestation, that is after the infection. If we introduce these immunoglobulins, we can reduce the manifestation of the disease. Like, for example, diphtheria. Because it produces the toxins may be circulating in the body. Now, these toxins are effectively dangerous, so we can remove the toxins by introducing immunoglobulins against the diphtheria toxin. That is why passive immunoprophylaxis can be done. Example would include diphtheria, tetanus. For example, a person has developed tetanus. Then what we need to do? We have to vaccinate, that is fine, but for the treatment purposes, we need to give the immunoglobulins. Again, varicella zoster, CMV, rabies, hepatitis, etc. One more concept I will just touch upon, that is the concept of cold chain. You will learn this more in your PSM classes. What is the cold chain? Please remember all the agents which are used in immune prophylaxis, whether it's a vaccine or immunoglobulin, whatever it is a biological material. So any biological material is prone to degradation. The two main aspects of degradation is high temperature and prolonged duration of storage. The cold chain is how we must keep these biological materials from production to introduction to the human body. So these are produced in a factory. From there, it is sent out to the wholesalers, to retailers, and then from the retailer to the hospital or the healthcare system where people are vaccinated. So throughout this journey, we must ensure that these biological materials must be stored at an optimal temperature and only for a particular duration of time. If we can maintain that, that is called as the cold chain. Furthermore, if we look at any vaccines, all vaccines will have this kind of a figure on their bottle or ampoule. These are called as the vaccine vial monitors. One look on this image will give you an idea whether we can use the vaccine or not. Normally, you can see an inner square inside the circle. Now, if the color of the inner square is lighter than that of circle, you can use the vaccine. If it is similar or darker than the outer circle, that means this particular bottle or ampoule has been exposed to higher temperature or has been kept for a prolonged duration of time. So it cannot be used. This is called as the vaccine vial monitor. Okay, fine. The last part that you must understand is the national immunization. Till now, I already have told you that immunization is a very effective method of preventing the disease in an individual and also to prevent spread of disease in the population. The government of India has already established a national immunization program for vaccination of children and pregnant women. This is the vaccination schedule that you must learn and you have to implement. I'll just go through it. At birth, that means whenever a child is born, within 72 hours, some vaccine should be given. It would include the BCG vaccine, oral polio vaccine, and hepatitis B birth dose. After that, at 6 weeks, these vaccines are given like the OPV1. The pentavalent vaccine means DPT plus Haemophilus influenzae B and hepatitis B virus, rotavirus, injectable poliovirus, and pneumococcal conjugate vaccines. At 10 weeks, OPV2, pentavalent, and rotavirus. At 14 weeks, OPV3, pentavalent 3, IPV, rotavirus, and pneumococcal vaccine 2. Again, the child is vaccinated at 9 to 12 months with measles, rubella, JE in areas where JE infections are seen, along with PCV booster. At 16 to 24 months, that is when the child starts to go to a preschool. At that point of time, the child has to be given with a booster dose of measles and rubella, JE in areas, DPT boosters, and OPV booster. Again, when the child at 5 to 6 years of age, when the child goes to the school, Then we have to give a booster of the DPT dose, and at 10 years we need to give the tetanus and diphtheria boosters again. Pregnant woman during the pregnancy, both diphtheria and tetanus vaccines are given. However, in this case, the dose of the diphtheria vaccine is reduced as compared to the child. So that is a low dose diphtheria vaccine that is given to pregnant woman. Now keep in mind, this is the schedule that has been recommended by the government of India. That means each and every child in India and a pregnant woman should get these vaccines. However, in our practical experience, we do get other subset of populations who need vaccination. For very common example would be us. We are the healthcare worker, even we are exposed to various diseases. We must also be protected. So, in healthy adults, what are the vaccines that can be given is a booster of the DPT vaccines. This is given because most of the time we as adults have been vaccinated in our childhood, and as we age, our immune memory reduces. So, a DPT booster has to be given. MMR, influenza, pneumococcal, human papilloma virus, which is a very common cause of, any idea? Cervical carcinoma. It infects both males and females, hence vaccination should be given to both males and females. And herpes zoster virus, that is the chickenpox virus. It may cause zoster infection in the adulthood. Finally, some people, because of their occupation, are exposed to some particular type of diseases. For example, a veterinarian who is handling dogs and cats has a higher probability of getting bitten by a rabid dog. So that person should be given vaccine prior to the exposure. Similarly, people working in the healthcare system are like us are very much exposed to diseases like hepatitis B, meningococcal, varicella, influenza B, typhoid, cholera. We are exposed, hence we should also be vaccinated. Another example I have told you about rabies. People who are handling dogs, veterinarians, some people who are involved with mass human migration, various fairs, etc. And in those areas, infections with cholera, typhoid are very common. Hence, in those particular subset of population, this vaccination can also be given to prevent an outbreak. So, to conclude, I think I was able to make you understand what is immune prophylaxis, how it works, what are the types by which we can prevent a disease, that is active immunization and passive immunization. In active immunization, we have the vaccines, the various types of vaccines, and in passive immunization, we can give immunoglobulins. And finally, there is a immunization schedule, there is a national immunization schedule, there are also other immunization schedule recommended by the WHO, CDC, etc. But in India, we have to follow the national immunization schedule. Okay.